work package 7 within MD-Paedigree is dealing with genetics, and in particular is addressing to three major areas of interest and research. The first area is dealing with cardiomyopathy, which is a quite heterogeneous disorder, which is due to multifactorial or Mendelian inheritance. In particular, we are testing in this project about 55 genes by using next generation sequencing technology, and the final goal of this research is to try to subclassify the patient affected by cardiomyopathy in subtypes in order that we can provide better genotype-phenotype correlations. The second topic is referring to obese children, in particular to the cardiovascular risk in obesity, and to reach this goal, we are currently investigating some dozen of gene variation which are known to be related in some way with obesity and with its complication. What we are doing now is try if we found some marker that can be useful to predict the risk to develop cardiovascular disease during obesity. The third other interest of this work package is most original in some respect, and is dealing in particular with gut microbiota analysis. Gut microbiota is particularly the whole of the microbes which are colonizing our intestine a few hours after birth, and during adult life we have about 1.3 kilograms of bacteria in our intestine, which are very important to modulate our immune response and also to modulate our genome.
During the MD-Paedigree project, we have a particularly focus on 3-4 different kind of diseases related to obesity, related to juvenile arthritis idiopathic, and related to disease, especially important in pediatric subjects. So we have performed from our bench work the mapping of all the genomes of the bacteria in these babies, and we have compared their genomes to the genomes from reference healthy babies. All these genomes, which are actually called metagenomes, depict the entire picture of the microbiota, and so produce the traits which are actually defined profiles of microbiota. So in this way it's possible to generate different profiles and to correlate these profiles to reference healthy profiles and see what is actually different in the different diseases.
One critical point of the gut microbiota analysis was to be able in some way to express the results coming from 100,000 of information that are derived from the study in something that can be reported and can be clinically useful. And we succeeded in this, and we for the first time were able in this study within the MD-Paedigree project to develop an original method which is mathematically analyzing these results, and in particular we express the result on a logarithm scale.
So based on this possibility, on this project, we have developed several technological tools. We have put together with the clinicians all the knowledges coming from clinicians, and we have also performed a highly technological platform aiming to produce big data related to microbiota in terms of description of genomes, in terms of description of metabolites. For example, we have established with the clinicians the possibility to modulate, to improve improve microbiota associated to disease by using, for example, probiotics, by using modulated diet, in a way to revert some microbiota, some altered microbiota into physiological microbiota. So this can be our descriptive approach, of course, but is also becoming a very strategic and functional tool to assist in the management of the diseases and to provide some laboratory medicine tools, but also some clinical medicine methods. The highly interactive way to perform MD-Paedigree is actually provide us the right way to produce some strict tools to assist the clinicians in the management of important diseases. And this can actually open the way to a new way to do medicine, which is actually defined as systems medicine, which actually can overall change the concept of medicine during this period.